Then it's time to introduce you our today's speaker. It's for me a pleasure to introduce you Julia Chocarro, that you can see on the screen. Julia is a biomedical engineer, graduated in 2019 from the Carlos III University in Madrid. After that, she performed an internship of 9 months at the 3B's research group in Portugal. During the pandemic, she complemented her studies with a master's degree in biostatistics and bioinformatics from the Open University of Catalonia. And since then, she's been working as a PhD student at the Center for Applied Medical Research in Pamplona, under the supervision of José Luis Lanciego. Currently, she's in her second year of the PhD program, and her project aims to develop and characterize a new non-human primate model of Parkinson's disease based on the time-dependent accumulation of neuromelanin in the substantia nigra. Julia's talk today is entitled Blaming Neuromelanin for Parkinson's Disease. Time-dependent tyrosinase overexpression drives endogenous synucleinopathy in non-human primates. So Julia, welcome. Thank you very much for your willingness to share with us your work. And you have the floor. So please go ahead and share the screen and go with your presentation.
Wait, sorry.
Put it in full screen, please.
Oh, sorry.
Okay.
Wait. Something went wrong. Full screen.
Yeah.
Like now?
Okay. Yeah. Nice. Fine. Thank you.
Okay. Thank you, Miguel, very much for the presentation. Good afternoon. My name is Julia. As Miguel already introduced me, I will skip this part. Well, first of all, I would like to make a brief description of what's going on Parkinson's disease, even though many of you, I know that you are already aware. Parkinson's disease is characterized mainly by the neuronal death of the dopaminergic cells of the substantia nigra, which provokes a dopaminergic depletion in the striatum. This impairment of the nigrostriatal pathway will eventually affect the motor system and give rise to the commonly known clinical picture of the disease. The dopaminergic neurons of the substantia nigra, as the name implies, contain a dark pigment in their cytosol known as neuromelanin. As the cell dies, they release this product to the extracellular space, which will activate the immune system of the brain and will try to eliminate it through the blood vessels. Finally, and most importantly, the neuropathological hallmark of the disease is the presence of the known Lewy bodies, which are some kind of cytosolic inclusions made of misfolded alpha-synuclein. Well, with this in mind, what are we lacking in the existing animal models? Well, first, in order to design the perfect animal model, we need to have in mind exactly what we want to reproduce and how we want to reproduce it. Most of the actual models, they administrate exogenous proteins or toxins, and they will accelerate this neuronal death of the dopaminergic cells to an extent that the model has no time to form the Lewy bodies that, as I have mentioned, they are the main hallmark of the disease, the neuropathological hallmark of the disease. Therefore, we think that we need to reproduce exactly and mimic what's going on in the brain of a Parkinsonian patient, and therefore we need to have these Lewy body-like inclusions in our model. As well, there must be a progressive neuronal death caused by the accumulation of alpha-synuclein. In this way, we will increase the therapeutic window and will enable testing of many more disease-modifying therapies. We wanted, indeed, to develop a new animal model that fits what I've just described. And to do so, we focus our attention on neuromelanin. But why neuromelanin? Because think about it, the neurons that are affected by the disease are the ones containing the pigment. Also, we as humans are the only ones who not only suffer from the disease, but also who accumulate neuromelanin to this extent, to the extent that we see really dark our substantia nigra. It appears at the age of 3 years approximately and accumulates throughout our lives. As said so, the time has an important role in the development and the pigmentation of the substantia nigra, and definitely so it does in the onset and evolution of the disease. Not only that, but only pigmented neurons in the substantia nigra exhibit intracellular inclusions of alpha-synuclein, the Lewy bodies mentioned before. With all this in the works, we designed the following strategy plan. Thanks to the vector sent by the group of Dr. Villa, we planned to inject an adeno-associated vector coding for human tyrosinase into the substantia nigra of 4 monkeys, 2 males and 2 females. We injected in the left substantia nigra of the monkey, and we injected the control vector on the right one. Tyrosinase, being the enzyme catalyzing the production of melanin in humans, was supposed to transfect the dopaminergic neurons and make them express neuromelanin and progressively accumulate it in the cytosol of the cell. Since the cell lacks the mechanism to eliminate this pigment, this accumulation will make the cell aggregate the alpha-synuclein into the known Lewy bodies. At some point, due to this Lewy body pathology, the cell will die and it will release all the pigmented material, the neuromelanin, to the extracellular space, causing a pro-inflammatory scenario. The cells of the microglia and macrophages will come, take the neuromelanin, and try to eliminate it through the blood vessels. Well, this was the plan. Let's see what happened. First, after the surgery and before the sacrifice, we could keep track of what was really going on the brain of this monkey, thanks to some neuroimage techniques. As for example, in the first image, we can see the MRI sequence, structural sequence, and we can see there's 2 injections right into the substantia nigra. In this other image, however, we used a sequence for neuromelanin, and as you can see on this behind this red circle, there is an hyperintense area, which means that there has been an accumulation of this neuromelanin. There has been an overexpression of the pigment. At the same time, every 2 months, we have done some micro PET images, DTB set, which shows how active is the striatum in terms of dopamine uptake. And as you can see in all of the animals, we have the basal image in the first row, and then every 2 months we did another one. And in all of them, we see some dopaminergic depletion in the left side. 2 of the monkeys, the M308 and M310, were sacrificed 4 months after surgery, while M309 and M307 were sacrificed 8 months before. The differences between both sides of the striatum of the putamen and caudate was statistically significant only in the females. We still don't know why, but could be due to could be a matter of gender or just due to serendipity or stereotaxic delivery. After the sacrifice, we found the first evidence of neuromelanin accumulation in the left substantia nigra of all the animals to its whole extent, from the most rostral level to the most caudal one. I've just shown you 2 animals, but we could see them in 4 of them. With this image as well, in the top one, there's the female sacrificed at 4 months and the other female sacrificed at 8 months. I want you to show that there is a difference in the intensity of the darkness of the substantia nigra, which could be, we suppose it is, due to the neuronal death caused by this accumulation. Regarding histology, first we counterstain the substantia nigra with neutral red to see the pigmented neurons at a micro level. You can observe the blood vessels here in this one, in the 7 number picture, that the blood vessels are surrounded by a high content of neuromelanin that probably is due to all these inflammatory cells that have taken the extracellular neuromelanin and take them, eliminating them through the blood vessels. Well, the percentage of transduction is shown in this figure in the upper one, which goes up to 50% in one of the primates. Nevertheless, we are expecting to improve these numbers for the next experiments, making some arrangements in the delivery and the type of AB used. With this other image, this other figure represents the optical density of the intracellular neuromelanin at a single cell level, which, as you can see, are equally distributed and had a similar average intensity number of neuromelanin. Also, in addition, I wanted you to show this an example of how are we measuring this percentage of transfection. Indeed, this is an online platform named Euphoria, which is made of artificial intelligence. And in this example, we can see how the algorithm that we design is working out. How can the algorithm differ from the neurons that are expressing neuromelanin from the ones that aren't transfected? This helped us a lot, saving some time to do all the calculations. In this other image, we wanted to as well to prove that there had been a dopaminergic loss in the origin, that is in the substantia nigra. That's why we immunostain the substantia nigra with tyrosinase hydroxylase. And indeed, we found out that there had been that half of the cells died. Also, we wanted to check it with the destiny of the substantia nigra of the nigrostriatal pathway, which is the caudate and the putamen. This is just only an example from the most rostral level to the caudal one, and see how the caudate and putamen of the left side has less marker, meaning that there has been a depletion, a dopaminergic depletion as well in the destiny. Next, we switched to the immunofluorescence staining, and we could observe for the first time that there were intracellular inclusions positive for alpha-synuclein in the green channel, and as well for the P62 in the red channel, which, as you may know, it's a well-known marker for Lewy bodies. These inclusions always had appeared in the cells that were positive for tyrosinase hydroxylase in the blue channel, and all of these neurons had contain some neuromelanin, as how they all colocalize. To make sure that we were watching was a Lewy body-like inclusions, we also used a marker for ubiquitin, also known for marking the Lewy bodies in the human brains. And indeed, we found out that these inclusions positive for P62, they colocalize with the ubiquitin marker here in the green channel. One more time, you can see the neuromelanin inside the cells and the merged channels. With these other images, I wanted to point out that it wasn't a difficult task to find one of these Lewy body-like cytosolic inclusion, since, as you can see in this lower magnification of the image, there is a relative abundance of these inclusive positive for P62. All of them, again, one more time, in cells that were positive for T8 and that contain neuromelanin. There's in this figure, I'm showing you a higher magnification of these intracytoplasmic aggregations, positive for alpha-synuclein and P62. But I wanted to point out that this ring-shaped structure that resembles a lot the actual Lewy bodies seen in the Parkinson brain of a patient. Also, it is interesting noticing that in the bright field here in the fourth column, you can already see like there's a protein aggregation which differs in density from neuromelanin, but still you can see it in the bright field, and it's always inside this ring-shaped structure positive for P62. Finally, we wanted to prove that there was as well the characteristic pro-inflammatory scenario, very typical from a Parkinsonian brain. And to do so, we immunized our tissue with IBA1, a marker for activating microglia, and another marker for CD62 for macrophages. They all colocalized, and actually, if you can see in this higher magnification, those cells are containing neuromelanin, but are not positive for Th marker. So indeed, we believe they are taking the extracellular neuromelanin and taking them to the blood vessels so they can eliminate it. So I know this was a lot. I wanted to make a recap of what we have seen in our Parkinson's disease model in non-human primates. So first, we saw a progressive dopaminergic lesion in the striatum observed first in the PET images, in the micropate. After sacrifice, there were macro and micro evidences of neuromelanin accumulation in the substantia nigra. Also, a macroscopic evidence of dopaminergic cell loss, both in the origin and destiny of the nigrostriatal pathway. Most importantly, we observed the presence of Lewy body-like inclusions positive for P62, alpha-synuclein, and ubiquitin. Finally, we proved that the presence of a pro-inflammatory scenario. So with all of this, we're happy to say that we are getting closer and closer to develop and characterize a new PD model in non-human primates that really mimics the known neuropathology of the disease, and that will therefore narrow the gap between animal experimentation and clinical trials. Well, as I've said, even though there were a few things that need to be addressed in order to improve this model, we believe it's quite a good model. However, we think the main message, the take-home message of this whole presentation is that we have evidences that prove that the neuromelanin accumulations is the one that triggers the endogenous synucleinopathy in dopaminergic neurons of the substantia nigra. Well, leaving that on one side, we wanted to address another issue, which is Bragg's hypothesis. I know that you may already be aware of it, but briefly, he proposes and his colleagues that the disease propagates in the brain caudal-rostrally from the lower brainstem through the midbrain, where is the substantia nigra, and forebrain, and into the cerebral cortex. He hypothesizes that first the disease initiates in the periphery, getting access to the central nervous system and affecting some susceptible areas as the severity of the disease increases. The way this alpha-synuclein, the Lewy body inclusions, the way they propagate is by permissive templates to the alpha-synuclein of the other area. In other words, we could say they suggest that the alpha-synuclein transports in a prion-like way. So we thought, since our animals had a considerably long follow-up, 8 months, let's check the prefrontal cortex, and there it goes. We actually found inclusions which were positive for P62 and alpha-synuclein in the pyramidal cells innervated by the dopaminergic neurons of the nigra, supporting Bragg's hypothesis that the alpha-synuclein spreads in a prion-like manner. In this image, we can see a neuron from the cortex positive for NUN in the superior frontal gyrus. Well, it's receiving a dopaminergic innervation proved by the fiber in the blue channel positive for T8. And at the end of the fiber, it's visible an inclusion positive for P62. We have many other examples, as you can see in these other images, that instead of neuen, we used DAPI to mark all the cells in the cortex, all the pyramidal cells in the cortex. And as well, we used alpha-synuclein marker to see if they co-localize with the P62. All of them had an innervation, possibly for TH, coming from the substantia nigra, the dopaminergic neurons. And in the last channel, we see all these channels merged together. These examples were seen in the superior frontal gyrus, but also in the presentral gyrus and anterior cingulate gyrus of all of the animals, not only just in the examples that were showing you. Well, these studies have already been done, but always using some exogenous alpha-synuclein. So this could be the first time that the accumulation of neuromelanin in the dopaminergic cells has triggered the formation of alpha-synuclein and P62 positive inclusions in the pyramidal cells of the cortex at different levels, suggesting that the alpha-synuclein spreads in a prion-like manner. Well, coming to the end, as I have mentioned many times before in the presentation, we are still lacking some things in our model. That's why we moved forward already with our work. So within the ongoing experiments, first of all, we switch from the AV1 to the AV9 because this vector is a better option for central nervous system application, as we have proved before. Also, the vector was delivered to the putamen instead of the substantia nigra to gain access to this substantia nigra by taking advantage of the retrograde spread of AV9. So we could say that the substantia nigra is kept, it's not touched. Like everything that we're doing it by injecting in the putamen. So any of the inflammatory response that may happen is going to be due to the accumulation of neuromelanin instead of to the injection. Also, these experiments, we increased the N. We used 8 animals. 4 of them were females and 4 of them males. Within these groups, 4 of the animals were injected unilaterally, again, as the ones that I have shown you during the whole presentation. And other 4 monkeys were injected bilaterally with the human tyrosinase. So, why are we injecting bilaterally? The reason is that we want to know how long we have to go in time in order to induce a motor phenotype, because now with monkeys with just a unilateral injection, it is impossible to see anything since the other periphery of the brain compensates the loss of dopamine. So with a bilateral injection, we might have a chance to see a motor phenotype. So here I can show you like our first preliminary results given by the micro-PET images. In the first row is the basal image done before the surgery, obviously. Then we have the pictures taken 2 months post-surgery and 4 months post-surgery. These are the axial projection and coronal views of 2 animals. One of them, the first one, the M401, was injected unilaterally, and you can see how in the left side we are missing some uptake of dopamine, meaning that there is a dopamine depletion already at 2 months and more significance at 4 months. On this other side, on the right, we have an animal injected bilaterally, and indeed at 4 months, the uptake of dopamine in the striatum, it's really different to the one at the basal time, meaning that there has been a big damage on the innervation of dopamine into the striatum. Finally, this week, we have sacrificed 4 of our animals, 2 males and 2 females, 2 of them injected unilaterally and 2 of them injected bilaterally, and see what was really going on the brain, like at a macro level. And we had a big surprise when we were cutting them and see how on this image on top, it's a female with a unilateral injection. As you can see, to the whole extent of the substantia nigra, it's pigmented. Not only that, but the older female injected bilaterally at 4 months after surgery also express high content of neuromelanin in both sides. And indeed, I haven't added the results for the males yet, but we can see that the females, as I have explained you before with our preliminary results, they accumulate better the neuromelanin than male, which we still don't know why. Anyways, we are now working with the histology techniques, and we didn't have time to add them to the presentation, unfortunately. But, well, thank you very much for your attention. That was all. I hope it wasn't too much. I will be happy to respond to any question you may have, so please go ahead.
Okay, so thank you very much, Julia, for an excellent presentation. So now the paper is open for discussion. So as I said earlier, if anyone is interested in posting a question, please just raise your hand using the application, or send me a message through the chat option stating your name and your interest in making a question, and I will give you the floor to do the question yourself. So there is no need for you to write down the question. So while we receive the first question, let me also remind everyone that this seminar is being recorded, and the recording will be available at the CIBERNED website in the next few days. And also, if anyone is interested in upcoming webinars, there is a list of webinars for the next few weeks until the end of the year, also available through the CIBERNED website. Okay, so questions?
No questions.
Questions? Don't be shy. I don't see any hands being raised or any questions coming through the chat. Yeah, no, sorry. That was Julia. So any questions for Julia? There is some comments on the chat, but no questions.
Well, Miguel,
Yes.
I just want to have the chance to say that just in case any CIBERNED group is interested on pushing forward this model in non-human primates or in rodents, feel free to contact us or Miguel. We are more than happy in providing the AV to anybody that might be interested. We are using non-human primate, but we know it's going to work very nicely in mice, in rats, or whatever. So just in case there's anybody else who wants to test this AAV for any given experiment, we are more than happy in providing you with the AAV. That's for sure.
Okay, thanks, Lancy. There is a question coming through the chat. Maybe someone doesn't have a microphone and just wrote down the questions.
Yeah, the differences.
Can you see it, Julia?
Yeah.
Okay. From
yeah.
Garrido.
Yeah, thank you for the question. Actually, when we switched to the confocal microscope and see all these aggregated alpha-synuclein, we don't see any difference in the number of bodies between the males and the females. There are a lot in both of them. We haven't counted them, but we know for sure there are quite a lot in both genders.
Okay. Thank you, Julia. There is a question from Javier Fernandez Ruiz as well.
Well, yeah, definitely it can be done in mice as well. Definitely.
Any more questions?
If you want to get in touch with us, you can reach me through my email or of Dr. Lanciegos.
Okay, I see a hand
hands up.
raised by Ariadna Laguna. Ariadna, please go ahead.
Hi, thank you, Julia. Thanks for a nice presentation. I was wondering about the inclusions you find in the cortex or in the cortical areas. I couldn't really see in your images, but do you think the inclusions are actually in the dopaminergic projection, or are they actually in the new and positive neuron that is in contact with this projection?
It's inside the neuron, possibly for neuen or DAPI, but it's at the end of the innervation, possibly for TH. Yeah, it's like right when it ends, it appears the both inclusions.
Okay, well, but the inclusion is in the neuron itself.
Yeah,
and maybe I missed it, but was this inclusion also positive for synuclein and ubiquitin?
Yeah, we haven't tried it yet for ubiquitin, but for alpha-synuclein, they always co-localize.
Okay.
Yeah.
Thank you very much.
You're welcome. Thank you for the question.
Right, thank you. Any more questions? I don't see any.
I don't see any
hands being raised or questions coming through the chat. Let's wait for a few seconds just in case someone is still pressing. Okay, so this is the last call for questions. Okay, so I don't see any questions, any requests coming through the chat or any hands being raised. So I guess that is then time to close the session. And not before thanking Julia for a very nice presentation. You actually made it through it brilliantly, I would say. So congratulations. Thank you everyone for attending. And I hope to see you in one of the next webinars. Okay. So thank you again, Julia, and have everyone a great afternoon. Bye-bye.
Thank you very much for having me. Bye.